Yes, we live now. Welcome to Let's Learn Bio by NCA. This is Nirupadesh Amdiyal. In this session, we are going to discuss about microbes in human welfare. Within this, the first concept, introduction and microbes in household products. The first question, read the following four statements. Find out the correct option with respect to lactic acid bacteria. They produce acids that coagulate and partially digest milk fat. Milk fat, this is wrong. They digest and coagulate milk protein. They require suitable temperature for their multiplication. Yes, this is correct. They play beneficial role by checking disease causing microbes in our stomach. Yes, correct. They improve nutritional quality by increasing the amount of riboflavin. This is again wrong. Increasing the quality of milk by increasing amount of vitamin B12. So the correct statement or option, that is B and C. Option third is correct. Next question. In the formation of dough and cheese making, the main gas produced is, the both process are fermentation. During fermentation, carbon dioxide gas is produced. Methane and H2O, methane CO2 H2O, carbon monoxide, actually methane, hydrogen, carbon dioxide produced during sewage treatment, that is in anaerobic sludge digestion, methane gas production, biogas production. Here, during fermentation, carbon dioxide is produced. Carbon monoxide is not formed during any process because it is a toxic to the microbes. Next question. The vitamin whose content increases following the conversion of milk into curd by lactic acid bacteria is, that is just I told, vitamin B12. Next, the microscopic proteinaceous infectious agents are called, proteinaceous infectious agents are called prions. Viruses are nucleoprotein infectious agents consisting nucleic acid and proteins. Here, prions, they lack nucleic acid, only the abnormally folded proteins called prions. Viroids are only the nucleic acid infectious agents. They causes disease in plants only, viroids. Next question. Complete the following analogy. Swiss cheese, Propionibacterium shermani. Swiss cheese is ripened by using Propionibacterium shermani. Bread, it is ripened or produced by Saccharomyces cerevisiae, the yeast. Clostridium botulicum, it is used in production of butyric acid. Lactic acid bacteria, lactic acid fermentation. Candida albicans, that is parasite on mustard, candidiasis in animals also. Next question. Microorganisms are present in soil, water, air, bodies of plants and animals, thermal vents. Yes, in all of the above. In all these uh, conditions, microorganisms are present. That is, all of the above is correct. Next. Inoculum is added to milk during curd formation in order to coagulate milk proteins. Inoculum is the small amount of curd is added to the fresh milk called inoculum, consisting millions of lab. They multiply at a suitable temperature, produce acid. That produced acid causes coagulation of milk protein, and partial digestion of milk protein leads to formation of curd. Next question. Identify the figure and find out the correct option. The given diagram, the first A, it represents the bacterial colony. Bacteria grown on the nutrient medium, that is a Petri plate consisting nutrient medium in which they produce colonies, bacterial colonies. And here also the same Petri plate consisting nutrient medium, it produce fungal colony. First option is correct. Next question. Look at the figure and select correct option. The given diagram, it is a adenovirus that causes respiratory infection in human beings. Third option. This is not a prokaryotic. Prokaryotic organisms, it includes Monera members, that is a bacteria. This is a nucleoprotein particle. Of course, virus, it is a nucleoprotein particle. It is capable of causing urinary tract infection. This is wrong. This adenovirus causes respiratory infection in human beings. This is not a fungal colony growing in petri dish. Bacteria never grown on petri dish or nutrient medium because they always require host cells. Means in vitro they are not possible to cultivate. Yes. Next question. Next concept, microbes in industrial products. Within that first question, the chemical substance produced by some microbes that can kill or retard the growth of other microbes are called the antibiotic. Antibiotics are the chemical substances produced by some microbes that can kill or retard the growth of other microbes. Antibiotics. The first antibiotic discovered was the penicillin, discovered by scientist Alexander Fleming. Next question. The large vessels for growing microbes on an industrial scale are referred as. The large sized vessels used for growing microbes on industrial scale are fermenters. Next question. Microbes are also used in the commercial production of certain organic acids. Butyric acid is produced by dash and lactic acid is produced by dash. The microbe Clostridium botulicum is used to produce butyric acid and lactobacillus used to produce lactic acid. That is, the first option is correct. Here aspergillus is used to produce citric acid. Acetobacter aceti, it is used to produce acetic acid. Next question. Which one of the following alcoholic drink is produced without distillation? Wine and beer. The two alcoholic drinks are produced without distillation. Here the first option is correct. Whiskey, brandy, rum, these are produced after distillation. Next question. Read the given assertion and reason carefully. Select correct option. Assertion. Ethanol is produced by fermenting malted cereals and fruit juice by yeast. Correct. Wine and beer are produced by distillation of a fermented broth. This is wrong. So here the first option is correct. Assertion is correct. Reason is false. Third option is correct. Next question. Read the following statements with respect to antibiotics and state true or false. First, antibiotics means pro-life in the context of disease-causing organisms. This is wrong. Antibiotics means the pro-life in the context of human beings, against in the context of disease-causing microbes. Waxman discovered streptomycin and coined the term antibiotic. This is correct. Full potential of penicillin as an effective antibiotic was established by Chain and Flore. Yes, correct. Penicillin was extensively used to treat American soldiers wounded in World War II. This is also correct. So, the first A is false, B true, C true, D true. Second option is correct. For the discovery, Fleming, Chain, Flore awarded Nobel Prize. Next question. Read the following statements and choose the correct option. Yeast Saccharomyces cerevisiae is used for bread making, beer making. Yes, correct. Saccharomyces cerevisiae, it is used in the production of beer in alcoholic industry, hence it is also referred as brewer's yeast. The same is used in the preparation of bread making in bakers, hence it is also referred as baker's yeast. Second, microbes, especially yeasts, have been used from time immemorial for the production of beverages like wine, beer, whiskey, brandy, or rum. Yes, this is also correct. After penicillin, other antibiotics were also purified from other microbes. Yes, correct. All of the above is correct answer. Next, antibiotics have greatly improved our capacity to treat the following deadly diseases except antibiotics are used to treat a plague, diphtheria, colicancy. Cancer, it is not possible to cure or treat by using antibiotics. Next question. Which drugs are administered before and after an organ transplant procedure? During organ transplantation, cyclosporin A is administered because it is a immunosuppressive agent. It suppresses recipient body immunity. Thereby it helps to accept the graft. Streptokinase, it is used as a clot buster to dissolve the clots formed in blood vessels in the patient undergone myocardial infarction leading to heart attack. Anabolic steroids, they are used to build the body in bodybuilders. Steroids. Next question. A patient brought to a hospital with myocardial infarction is immediately given, just I told, streptokinase. It is a clot buster. It is obtained from the genetically engineered Streptococcus bacteria. Penicillin, it is a first discovered antibiotic by Alexander Fleming. Statin, it is a blood cholesterol lowering agent obtained by yeast again, that is a Monascus purpureus. Cyclosporin A, that is an immunosuppressive agent obtained from Trichoderma fungus. Yes. Next, the microbes in sewage treatment and microbes in production of biogas. Within that, first question, wastewater treatment generates a large quantity of sludge which can be treated by. After treatment of sewage, large quantity of generated sludge it is subjected for anaerobic sludge digester. The flocks are the group of microbes, that is the bacteria associated with fungal filaments to form bacterial flocks in a aeration tank. Chemicals, chemical treatment, that is a tertiary treatment. Next question. The microorganisms involved in flock formation during sewage treatments are there aerobic bacteria and fungi. Anaerobic bacteria, they are present in anaerobic sludge digester. Possibly the methanogenic bacteria are the anaerobic members. Autotrophic bacteria, they are not present in sewage treatment. Usually the heterotrophic microbes, they are used in sewage treatment. Next question. Read the following statements and select the correct option. Statement A. BOD is the measure of amount of oxygen that would be consumed if all the inorganic and organic matter in 1 liter of water is oxidized by bacteria. Here, inorganic matter, it is not possible. Only the organic matter. Greater the BOD of wastewater, lesser its pollution potential. This is wrong. If the greater the BOD of wastewater, greater will be the pollution potential. Lesser the BOD value, lesser its pollution potential. So both statements are incorrect. Fourth option. Next question. Given below are three statements, each with one blank. Select correct option that fills the blank in the statement. First, formation of flocks in Formation of flux is observed during a dash. Flux formation takes place during a secondary treatment in a aeration tank. Second, the supernatant left after the sedimentation of sewage water during secondary treatment is known as the supernatant. It is a secondary effluent. Supernatant during a treatment, the two fractions were formed, one that is a sludge, another that is a transparent supernatant. Third, BOD is the measure of dash in wastewater. BOD is the measure of organic load or organic matter present in wastewater. So the second option is correct. Next question. State true or false for the following statements with respect to sewage treatment. The treatment of wastewater in a sewage treatment plant is done by autotrophic microorganisms present in sewage. This is wrong. Heterotrophic microorganisms are used, not autotrophic. During secondary treatment in a secondary treatment plant, BOD initially increases, then decreases. Yes, this is possible. At the initial stage, it has a high value of BOD. Gradually, it's going to decreases. Anaerobic digestion of activated sludge produces biogas, includes sludge digester. Yes, this is also correct. During anaerobic sludge digestion, activated sludge it subjected for oxidation digestion. During their digestion, they produce gas. That process sludge digester. A small portion of primary sludge is used as an inoculum to start another secondary treatment. Yes, from the settling tank, a small fraction of portion of sludge is used as a inoculum to start another secondary treatment. Correct. So the first option. Next question. The Ministry of Environment and Forest has initiated Ganga and Yamuna action plans to treat wastewater. Yes, correct. Prevent the discharge of untreated sewage into these rivers. So the first and second option both are correct. Prevent microbial treatment of disease. This is wrong. Next, pick the incorrect match regarding sewage. High BOD of water, it is more polluted. Activated sludge serve as an inoculum in aeration tank. Anaerobic sludge digester, aerobic flux. This is wrong. Mismatch. Physicochemical process, tertiary. Yes, chemical treatment, tertiary treatment. It is possible when the sewage containing industrial waste. Normal sewage, municipal sewage, only the two steps are sufficient, primary and secondary treatment. If it is contaminated with industrial waste, then go for tertiary treatment, that is a physicochemical process. Next question. The primary treatment of sewage involves primary treatment, it involves sequential filtration and sedimentation. During primary treatment, floating debris were removed by using sequential filtration, and a grit it is removed by sedimentation. Grit it is a soil with small pebbles. Next question. Read the following statements. Statement A. Activated sludge in a settlement tanks of a sewage treatment plant is rich source of aerobic bacteria. Yes, correct. B. Biogas is produced by the activity of aerobic bacteria and animal waste. Aerobic, this is wrong. Anaerobic bacteria. So the first statement is correct. B statement is wrong. Third option is correct. Next question. Read the following statements with respect to biogas production and state true and false. The type of gas produced depend upon the microbes and organic substrate they utilize. Correct. Certain bacteria that grow aerobically on cellulose material produce large amount of methane along with the CO2 H2O. Aerobically, no, this is anaerobically. Methanobacterium is a common methanogen. Correct statement. Next option. Methanogens are commonly found in anaerobic sludge during sewage treatment. Correct. So the first A is true, B is false, C is true, D is true. The third option is correct. Next, choose the incorrect statement with respect to biogas. It predominantly contains methane. Yes, correct. Biogas is the mixture of gases produced by methanogenic bacteria, majorly predominantly consisting methane. Other gases includes carbon dioxide, H2O, hydrogen sulfide, no oxygen. Second, its production involves aerobic digestion of organic waste. Again, this is incorrect. Anaerobic digestion, aerobic, this is wrong. It is also called gobar gas, definitely, because the gobar, it is used for the production of biogas, hence it is called gobar gas. Methanobacterium is common biogas producing microbes. This is also correct. So incorrect is a second. Next, the figure given below shows typical biogas plant in which of the following option in products labeled as A, B, C correctly identified. This is a biogas plant consisting. Here A is a digester, C it is a sludge. It means this is after the processing, the remnant sludge. Here the dung slurry itself is going to fed. Slurry it is consisting equal mixture of dung and water. And B it is a gas holder consisting carbon dioxide, methane, carbon dioxide, etc. This is a pipeline connected to the pipeline of house outlet. The typical biogas plant consisting one inlet, two outlets. One the gaseous outlet, another that is a sludge outlet. Here the first A is a digester, B is a gas holder consisting methane and carbon dioxide, and C is a sludge. So the second option is correct. Next, methanogens do not produce. Methanogens do not produce oxygen because they are not aerobic. Methane definitely, hydrogen sulfide, carbon dioxide. These are the mixture of methane gas. Next question. Which of the following term is not associated with the typical biogas plant? Not associated. Concrete tank, 10 to 15 feet deep concrete tank consisting outlet. Yes, one gaseous outlet, another sludge outlet. Floating cover. The floating cover gas, typical biogas plant has a gas holder. It is having a floating cover. It's keep on rising as the gas is produced during a anaerobic digestion. Trickling filter, this is not found in biogas plant. So option 4 is correct. Next question. Find out which of the following statement is incorrect with respect to biogas plant. Again incorrect. It has a floating cover that keeps on rising as the gas is produced. Correct. It consists of 10 to 15 feet deep tank in which bio-wastes are collected and slurry of dung is fed. Yes, this is also correct. Spent slurry may be used as a fertilizer. Yes, definitely. Main gases produced are methane. Carbon monoxide is not produced. Nitrogen oxide. So, fourth option is wrong. Next question. Read the following statements with respect to activated sludge and select correct option. After secondary treatment, microbial flocks are allowed to settle down, which is called as activated sludge. Yes. The major part of sludge is pumped into large tanks called anaerobic sludge digester. Yes, definitely. It is generally released into natural water bodies like a river and streams. See here, it is not generally released. After treatment only, it is released into natural water bodies. So, option A and B are correct. C is wrong. The third option. Yes. Next, microbes as biocontrol agents and microbes as biofertilizers. Which of the following organism is not used during biocontrol method? The Monascus purpurus is not used during biocontrol method. Bacillus thuringiensis, Trichoderma, ladybird beetles. Three are used as a biocontrol. This is used in production of statin. Second, nucleopolyhedrovirus are. They are effective against insect and other arthropods. Fourth option. These are the narrow spectrum, not a broad spectrum. They are effective against rodents. This is wrong. They are effective against arthropods and insects. They belongs to baculovirus. These are the baculovirus. So the fourth option. Which of the following is incorrect about a baculovirus? Baculovirus are pathogens that attack insect and other arthropods, correct? Species specific insecticidal application, correct? These are not a broad spectrum, they are narrow spectrum insecticidal application. No negative impact on plants, mammals, birds, fish, and even on non-targeted insect, correct? So third option is incorrect. Next, which larvicidal fish is used to control mosquito larvae? That is Gambusa. It is used to control mosquito. Next question. Which of the following is an example of carrying out biological control of pest or disease causing microbes? Bt spores to increase sugarcane yield. Wrong. Nucleopolyhedrovirus against mammals, birds, and fishes. This is wrong. Ladybird beetle against mosquito. This is wrong. Ladybird beetles used to control aphids. Dragonflies used to control mosquitoes. Trichoderma species against certain plant pathogens. This is correct. Next, go through the following matches with respect to biocontrol agents and select correct option. Ladybird beetle aphids, correct. Dragonflies mosquito, correct. Bacillus thuringiensis butterfly caterpillar, correct. Trichoderma plant pathogen, yes, all of the above. A, B, C, D are correct. Next, match the items column 1, column 2, column A, column B. Ladybird aphids, Mycorrhiza glomus, biocontroller Trichoderma, biogas methanobacterium. So the second option. Next, identify the correct statement. Baculovirus are the pathogens that attack insects and other arthropods, correct. Nucleopolyhedrovirus are excellent candidates for species-specific narrow-spectrum insecticidal application, correct. Bacillus thuringiensis are available in sachets as dried spores which are mixed with organic solvent. This is wrong. They are mixed with water and sprayed into vulnerable plants. Trichoderma species are free-living fungi that are very common in root ecosystem. So here option A, B and D are the correct. C is the wrong. Next question. Microbes used to control pests under IPM includes dragonflies used to control mosquitoes, ladybird to control aphids, trichoderma also used. Here cactoblastis it is not used. Next question. Consider the following four statements related to organic farming. Select the correct option. True and false. The pests are not eradicated, but instead they are kept at a manageable level by simple system of check and balance within a living, vibrant ecosystem. The more variety landscape has, the less sustainable it is. Bioinsecticides and biopesticides are much better option than chemical insecticides and pesticides. Organic farming involves the use of only the chemical fertilizers. First, A true, B false, C true, D false. Second, read the following statements and choose a correct option. Statement 1. Bacillus thuringiensis are available in sachets as dried spores which are mixed with water and sprayed into vulnerable plants such as brassica, fruit trees, where these are eaten by insect larvae. In the gut of larvae, the toxin is released and the larvae get killed. Yes, the second statement. The bacterial disease will kill the caterpillar but leave other insects unharmed. This is again correct. So the both statements are correct. Next, an organism used as a biofertilizer for raising soybean crop is, that is a rhizobium. It is a symbiotic association with the soybean. Azospirillum, free living nitrogen fixer. Frankia, it is also symbiotic bacteria associated with non-legume plants. Anabina, it is a cyanobacteria, nitrogen fixing cyanobacteria. So here second option is correct. Next, biofertilizers are microorganisms that bring about soil nutrient enrichment. Biofertilizers are the microorganisms that increases soil nutritional quality. The first option. Next, which one of the following is not a nitrogen fixing organism? Anabina is nitrogen fixing, Nostoc is also nitrogen fixing, Azotobacter is also nitrogen fixing, Pseudomonas it is a non-nitrogen fixing organism. So the fourth option. Next, read the following statements and choose the correct option. In a non-leguminous plant, rhizobium fixes atmospheric nitrogen into inorganic form, which is used by plant nutrients. In a leguminous, not in a non-leguminous. So the first is incorrect. Second, the fungal symbiont in mycorrhiza absorbs phosphorus from soil and pass it to the plant. This is a correct. Second, there are problems associated with overuse of chemical fertilizers. There is a large pressure to switch to organic farming, not inorganic. This is again wrong. So the third option, all statements are incorrect except two. Second option is only the second statement is only the correct. First and third are incorrect. So the third option is better for correct. Which of the following are free living nitrogen fixing bacteria found in soil? Free living nitrogen fixing bacteria, Azospirillum, Azotobacter are the free living forms. Rhizobium, Frankia are symbiotic. Azolla nostoc, they are also associated with other organisms. Frankia oscillatory also. So free living nitrogen fixers, Azospirillum, Azotobacter. Biological control of a pest is, they are self-perpetuating. They are not causing any pollution. They are not again expensive. They are not toxic. Third one is a correct option.